Okay, so the title of the poster is Chemoresistance Characterization using Raman Spectroscopy. So this is a pilot study data that I am presenting today. So one of the most important challenges that we have during the cancer therapy is the presence of resistance or incidence of resistance that patients report. So the resistance can be either in form of chemoresistance or as radio resistance. So it's a serious pitfall in the treatment of cancer. And therefore, if we can develop some strategy to early detect and predict these incidences, they can be prevented, or we can also devise new strategies to avoid the incidence of resistance. So there are several methods that people explore to understand and develop strategies to do early prediction of chemoresistance. And one such method is Raman spectroscopy. The advantage of using Raman is that it's a rapid label-free technique. And it's an inexpensive technique which provides biochemical information of the sample of interest. So it can be a cell, biofluid, anything. So in this study, we have taken two different cancer models, cervical and liver cancer. And we have taken two different cell lines of each of these cancer models. And so once we have developed the resistant cell lines against a doxorubicin drug, which is again a common chemotherapy drug that is used in clinics. So we have the parental resistant cell line. Then we fix the cells and we pellet down and we record the Raman spectra. So around 10 spectra is recorded per cell pellet, and then each of the spectral data is subjected to the pre-processing methods such as smoothing, baseline correction, normalization, and then it is subjected to multivariate analysis. It is PCA and PCLDA. So this is the mean Raman spectrum of cell line that we get. So we can see that the biochemical features such as proteins, lipids, cytochromes and nucleic acids are something that we get out of the Raman data. And we see that the proteins and the nucleic acid intensities are higher in the parental cell lines of all the cell types. And the resistant cell line, they show higher lipids and cytochrome related feature in the Raman data. After this, the mean Raman spectrum is subjected to multivariate analysis. So we had performed multivariate analysis of all the cell lines, and then so here I am showing a data of CH, which is a cervical cancer, and Hep3, which is a liver cancer model, and we can see that the method is efficient enough to stratify the parental and resistant population with a good accuracy. So in conclusion, we can say that we have explored this as an understanding to find out what are the chemoresistance associated changes that we can get, and whether there is any overlapping spectral signature that we can find out. And indeed we find that lipids and cytochromes are there in the are present higher in the resistant cell line. This shows that there is a potential of Raman in using this in clinics to stratify the patient population which are prone to resistance or which will develop resistance in future. And further validation of this is required using a larger sample size, as well as we need to understand using electron microscopy what are the subcellular changes and whether those subcellular changes are also overlapping in nature of their occurrence. Like in all the resistant cell lines, we are able to find out whether there are some mitochondrial associated changes or something else. And if we can get this kind of understanding, we can use that also and develop models in future with that, and that can be helpful in clinics. Thank you.